All right, so to kick off our last section of the day, Andy Lichtman's going to tell us more about CD4 and CD8 T cells.
Okay, so I have a real tough battle. Following a boule, I always feel boring and rushed. This is the typical procedure. But what we're going to do is, and I appreciate it if you're allowed to fall asleep now. A boule didn't want you to fall asleep, but I realize that it's been a tough, long day. My feelings won't be hurt. We're going to take it from where Boule left off. He really told you about how T cells get activated and that what some of the breaks are in activation, events that largely take place in organized lymphoid tissue such as lymph node or spleen. He told you about the exquisite molecular regulations of those processes, but he told you very little about what T cells actually do in the immune system, what their effector functions are. And so that's the baton that's passed to me to tell you about it. In previous versions of this lecture in previous years in this course, we ignored CD8 T cells entirely, which really is not very fair and not a very balanced view of the adaptive immune system. So I've added something about CD8 positive T cells. That means that I had to remove a little bit of detail about cytokines, which are the main theme for the CD4 part. So like everything in a course like this is a broad survey. Some things are sacrificed and there'll be questions that are not answered. And I'm erring, again, on the broad scope of things rather than drilling into too much detail in any particular aspect. So just to remind you, and a Boule gave you some of this, and in fact, we've heard this throughout the day, in terms of T lymphocytes, there are two large categories or subsets of T cells. The CD4 positive T cells, which we often call helper T cells, because really the way they work is they help other cells to do the business of eradicating infections. And so in this cartoon, we have helper T cells that I'll define these subsets later that are helping a macrophage that happens to be infected with a microbe to be able to do things that are useful. For instance, activate the macrophage so that it can kill the microbe that's inside, or to produce inflammatory cytokines that can bring more leukocytes to a site of an infection. And the second branch are the CD8 positive T cells that we call cytotoxic T lymphocytes, or CTL. And as you've heard several times, they directly kill infected cells. So we're going to spend the first part of the lecture talking about the CD4 positive helper T cells, and the second part, which is really not half but a third of the lecture, on CTLs. Remember, just because we're not going to go into a lot of detail, that the recognition of antigen by the CD4 positive helper T cell is with what type of MHC molecule? Class 2 MHC, so a peptide that's bound to class 2. And the CD8 positive T cell is recognizing a class 1 MHC associated peptide, and you heard in elegant detail about how these different peptide MHC complexes are generated and how the microbes may be in different locations. And this speaks somewhat to the type of infections that these two types of T cells deal with. And so as I deal with CD4 positive helper T cells and their function, I'm really going to focus the way I'm going to talk about it with the cytokines that they make. That's not the only way to talk about helper T cells, but that's what I'm going to talk about, and talk about subsets of these CD4 positive T cells that make different cytokines. And this idea that not all CD4 positive helper T cells are the same came many years ago, more than 20 years ago, when people realized that these helper T cells actually helped a lot of different responses, and it wasn't clear that one T cell could make all the cytokines that could do this sort of thing. For example, the first definition for help really was to help B cells make antibodies, and Shiv Pillai tomorrow will tell you about how helper T cells help B cells, but it was clear that cytokines were very important in that. And then it was also clear from classic experiments that helper T cells also made cytokines that activated macrophages to kill intracellular microbes. And even more we know now, there are helper T cells that promote inflammation and bring leukocytes into sites of infection. And there's been a long literature that's been a little bit murky about how CD4 helper T cells help cytotoxic T lymphocytes to actually do their job. And so the question is, how can one subset, basically, of helper T cells make all the types of cytokines that are needed to do all these functions? And the hypothesis was made that or stated that perhaps there are these functional subsets, CD4 positive helper T cell subsets. And so I'm not going to sort of bring you through the long history of how these subsets were discovered, but it was about 20 years ago and a little bit more, and it was two investigators, Mossman and Kaufman, working at a biotech firm who identified that the cytokines made by some clones of T cells, and a clone is a set of T cells that are all identical, derived from a single cell, would make cytokines X, Y, and Z, whereas another clone would make cytokines A, B, and C. And although there may be some overlapping cytokines, they're ones that define them as different. And so these T cell subsets were first defined that way, first in mice, in mouse experiments, and then in humans. And so there are populations of CD4 positive helper T cells that make restricted and non-overlapping sets of cytokines. The cytokines that a particular subset makes begin early after activation. These T cells make a lot of different cytokines, and after progressive multiple rounds of replication, for instance, in the events that Abul told you about, in that lymph node, when the naive T cell sees the APC peptide MHC complex and sees the co-stimulatory signals, it begins a round of differentiation and proliferation. And you need several rounds of proliferation before these T cells are actually polarized to be a subset. Each subset has distinct functions, largely based on the cytokines they make, but they also happen to do other things differently, such as they migrate into places differently, and their roles in diseases are different. And so I'm just going to jump right to the overview summary of what these subsets are before we return to each one and say, how do we get from a naive T cell to that type of subset? And then what these subsets actually do to protect us against infections and what they may do actually to cause disease. And so the best defined, the triumvirate of T cell subsets that we now talk about, and it's so prominent in the literature, are Th1 cells, Th2 cells, and here's another example of idiotic immunology nomenclature, Th17 cells. So the Th17 cell was not the 17th type of T cell subset to be discovered. The 1 and 2 just are the first two. So these two are not named after the cytokine, and this one is, and that's the way it's going to be, apparently. So Th1 and Th2 were those original two that I described, Mossman and Kaufman, defined first in mouse, and then we have it in human. So the best way to define these subsets are the cytokines that really define them, the signature cytokines. And Th1 cells, that signature cytokine is interferon gamma. By the way, we have not really had a formal definition of cytokines, but you've heard about them in every lecture so far, and we don't really have room to have a dedicated lecture on cytokines. Besides, that would be a very boring lecture. It would be like a table of different proteins. But these are proteins that really speak between cells. So one cell makes it in the immune system, and another cell responds. In fact, we know now that cytokines are made by non-immune system cells and talk to immune system cells and vice versa. And basically, they're proteins that act not really as hormones in the sense that most of their activity is right next to the cell they're working at. They work in sort of paracrine or autocrine ways. But these proteins can also be secreted in large amounts and have systemic effects. And Ken spoke of cytokines in the innate immune system, and cytokines certainly are very important for the regulation of the adaptive immune system. So here, interferon gamma is one of our key cytokines for effector functions, and it's made by these Th1 cells. And right to the chase, I'll get to these functions later, but perhaps its main function is to activate mononuclear phagocytes, in particular macrophages. It also has some effects on B cells and what type of antibodies they make. And it's a host defense role really is for microbes that are inside cells. Now, when I say intracellular microbes, that doesn't mean the microbe necessarily began there or lived there like a virus. A bacterium that is phagocytosed by a macrophage becomes intracellular. In order for that macrophage to be a really good killer of that bacterium, gamma interferon is important. Actually, the subsets we have much information about what they do bad as what they do good, and Th1 cells have been implicated as being important in a variety of autoimmune diseases, as well as a lot of tissue damage that may occur in what you think the appropriate response to an infection, but the bystander destruction of tissue that might occur is very much associated with Th1 cells. Th2 cells are a bit of an enigma about why we have them other than to make us miserable. because they're really the T cells that underlie allergic disease or atopy, the most common, and some people think the most important immunologic disease, of which in this room I would say 20% of people at least are affected by this. And they make the cytokines IL-4, IL-5, and IL-13, which have a variety of effects on cells that mediate the so-called immediate hypersensitivity diseases, in which mast cells become activated and release their mediators and cause hay fever, or allergic rhinitis, or asthma, and can cause severe reactions. And Steve Galley, on day 3 of this course, will be telling you a lot about that in detail, these parts of Th2 cells. In host defense, probably they've evolved because these reactions of the mast cells and eosinophils that are associated are good for ridding helminths or worm infections in the gut. And that may be sort of evolutionary pressure to evolve this branch of adaptive immunity. As I mentioned, allergic disease is the bad thing they do. Now, Th17 cells were only discovered, I don't know, is it now 5 or 6 years ago, not 20 years ago, with a tremendous amount of activity to catch up on the information for these, but now we know a lot about them. And the signature cytokine, as the name implies, is IL-17. Now, there are several forms of IL-17, and IL-17 is not only made by Th17 cells. Th17 cells make IL-17A and F. They make a lot more A than F, and most of the biologic activities between the two are the same. The receptors for these cytokines are ubiquitous. They also make a cytokine IL-22, which pretty much is unique to Th17 cells and not the others. And what they do in immune response is really promote inflammation. And in many tissues, except perhaps the brain, a lot of this inflammation has a lot of neutrophils in it. And so neutrophils are the hallmark, as Ken Rock started his first lecture today telling you about neutrophils being a component in innate immunity, and without neutrophils we all die. And you don't think of it as an adaptive immune response. We often say acute inflammation is innate immunity with a lot of neutrophils, and chronic inflammation with lymphocytes and macrophages is adaptive immunity. Well, here is a T cell that promotes a lot of neutrophilic inflammation. And as you might gather from the little bit you've heard about what neutrophils do, they're great eaters of bugs. They're phagocytic and they kill bugs because they make that Clorox. In fact, the role of this subset is importantly for extracellular infections with bacteria and fungi that really need neutrophil responses for control. And again, Th17 cells, like Th1 cells, have been implicated in autoimmune disease. By the way, I know that you want me to get done so you can go get your margaritas. And it sounds like maybe I'm pressured to get through this, but please interrupt with questions. If I can't answer them, I'll ask Abul to answer them in the back. So we have an interesting question here that really began with the Th1, Th2 subset, and it was a real enigma and a very interesting scientific problem. You start with a naive CD4 positive T cell, which I'll tell you on the outset, there's no evidence whatsoever that that naive T cell that comes from where? Where does the naive T cell emerge from? The thymus. Okay, it's a CD4 positive naive T cell. It has no predetermined predilection to becoming a Th1, Th2, or Th17. There's nothing in thymic development that says that this cell will ultimately become a Th1, Th2, or 17. This is something that happens in response to signals that that naive T cell gets when it first sees antigen presented by what type of cell? Dendritic cell, naive cells. They need dendritic cells. Those are the ones that activate the naive cells. So something about that, what happens when the naive cell presents antigen to the dendritic cell presents antigen to the naive T cell starts telling this cell to differentiate down one or another pathway. And so it turns out that each of these pathways, or each subset, tends to be induced by stimuli that at the end of the day get an effector cell that is pretty good at dealing with that type of infection. This is sort of the teleology we like to build onto this immune system, but it makes sense evolutionarily that the signals that drive to a Th1 cell will be the signals that are generated when you have an intracellular infection. And the signals that drive for Th17 would be those that are provided when you have an extracellular bacterial infection. Or the signals that drive Th2 would be theoretically signals you get from worm infections, although there we don't quite know what they would be. All right, so it turns out that the main driver down each of these pathways The most important, not the only one, but the most important one are the cytokines that are produced at that time of antigen recognition by probably the antigen-presenting cell itself, such as the dendritic cell, or other cells in that small microenvironment. Perhaps even cytokines that are made in a small amount by the naive T cell itself when it first sees the signals from the T cell receptor. And they feed back onto receptors on that naive cell and start a process of differentiation. And so the source of these cytokines has been an interesting puzzle for these pathways, but we have some clear definitions of what these cytokines are for certain subsets. So the main features of T cell subset differentiation are that they come in phases. The first we can call induction, and that's the time in the first encounter of the naive cell with the dendritic cell and the APC. And clearly the signals from the T cell receptor that Abul just told you about are essential to initiate the response. Co-stimulatory signals that you've heard an awful lot about are essential for the differentiation of any subset, and that goes hand in hand with the fact that you need both these, antigen and co-stimulation, just to start the T cell in its cycle of proliferation. Then there has to be some form of commitment down one or another pathway, these decisions are made very early at that process. During the first cell division, certain things start happening, changes in a transcription of genes, and then ultimately some more permanent changes that really amplify the system and cause commitment. So in amplification, there are various mechanisms by which the cell, which is now no longer a naive cell, but it's 3/4 of the way down to becoming a Th1 cell, will make things that reinforce Th1 differentiation, and the same for Th17 and Th2. And this is where we see the best examples of clear-cut polarization of subsets when we have a persistence of antigen and chronic stimulation and restimulation. So it feeds into this idea that you have more and more stimulation, you have amplification. I sort of throw that out. At what time do you think, what sort of diseases can we guess will happen where you never get rid of the antigen, or the antigen is hard to get rid of? Well, parasitic maybe is a good one, but if you had an effective immune response, you could theoretically get rid of the parasite. But if you live in an area that's endemic and you're always, getting contaminated, I suppose that's a good example. Well, that's a special case I won't get into because that's screwing up the whole process of CD4 differentiation. A type of disease where you'll never, ever get rid of the antigen that's driving the disease. Autoimmune disease, right? I mean, we're stuck with those antigens. And often the Th subset differentiation polarization is most obvious in certain autoimmune diseases. In fact, the mouse models that have driven looking at the human have first been, for instance, Th17 cells, autoimmune diseases, like the models for multiple sclerosis and inflammatory bowel disease. I was going to give the other example of allergies, which is also Th2. I mean, there are environmental antigens that, unless you move to Arizona, you're never going to get rid of. So, okay, so let's now go through a few of the details of how you drive differentiation of each pathway. There's a tremendous amount of elegant literature, including a lot of interesting transcriptional biology. And I'm not going to drill too deep into these, but sort of give you summary statements. So for the Th1 cell, remember, at the end of the day, you're going to have a cell that makes gamma interferon, the major effector functions of which are activating macrophages and perhaps driving some antibody differentiation by B cells. But you're starting with that naive T cell. So in addition to antigen, that is the peptide MHC binding to the T cell receptor, and the co-stimulation, I said that cytokines are important. And it turns out that a very important cytokine, both in mouse and human, that is needed for this process is interleukin-12. This is one of a family of cytokines in this particular family. It's a heterodimeric cytokine with 2 proteins on it. I mention that now because later I'm going to tell you about trying to block these pathways with antibodies so you can block one chain or the other. And IL-12 is known to be made by dendritic cells, but a resting dendritic cell, and we do studies in the lab, and a lot of people may in mice, you can take make dendritic cells or get them out of the spleen, and they don't make IL-12. You have to stimulate them. And I've given you the stimulus here, microbes. So what do you think is the actual mechanism of the production of IL-12 from the dendritic cell. What is the microbe doing? What possible receptor might it be binding to? Toll-like receptors, TLRs, that's right. So Ken Rock told you about toll-like receptors. One of the classic responses of toll-like receptor signaling in a dendritic cell is IL-12 production. For instance, LPS, lipopolysaccharide, or endotoxin binding to TLR4 on a dendritic cell will make it produce interleukin-12. So you can see that infections that have strong innate stimuli, that is, they are filled with, or bugs that have these pathogen-associated molecular patterns that bind TLRs, that activate these cells, will tend to drive towards Th1 responses. A second cytokine that is needed as well as IL-12 is gamma interferon. Now, there's a bit of a conundrum here. Gamma interferon is, I'm telling you, is needed for this process, but gamma interferon is actually the end product. So how do you get this gamma interferon? And here's where we have to evoke other cells, perhaps, besides the antigen-presenting cell. There's literature about the dendritic cell making all sorts of cytokines. Like, as Abul said, nothing is black or white or nothing or everything. And if you look hard enough, you'll find every cell making everything. But they're not abundant makers of gamma interferon, so where might it come from? And it turns out that there's a cell called the natural killer cell that Louis Lanier, we referred to Louis earlier, will be telling you about 2 days from now. And natural killer cells are innate cells. They don't need certain stimuli that I'm talking about here. They're present, they're ready to go, and they may respond, again, to toll-like receptors or to bugs to make gamma interferon. So that's one possible source for the gamma interferon here. So in Th1, and I'll show you this for Th2 and Th17, so what? The cytokines bind to receptors on the cell. In fact, there's a beautiful and intricate dance they play. In fact, for instance, the receptors for some of the cytokines are driven by other cytokines. So gamma interferon receptor may be induced by the IL-12 signal. So this initial signal allows the cell then to respond to interferon gamma. What the cytokine receptors do is they work through signal transduction pathways, which we're really minimizing in this course. They're an important and great literature. To date, there are relatively few therapeutic targets in these pathways. There are some, but perhaps that's why we don't emphasize them yet, because they've been hard to crack in terms of curing disease. There's some very important ones that have been established for a long time. Nonetheless, I'll just introduce you to a particular family of them, and those are the STAT proteins, or the signal transducers and activators of transcription. They are basically signal transduction molecules that become phosphorylated on tyrosines and move to the nucleus, and they bind to DNA, and they are part of the machinery that enhances transcription of genes. And so IL-12 has its particular STAT that it activates called STAT4. Interferon gamma has its STAT called STAT1, and both of these are needed ultimately for differentiation to Th1 cells. So in addition to the transcription factors that the cytokines directly involve, I'll just mention another, it's not a family, but another way of categorizing transcription factors that are important for T cell subset differentiation. And these are a name that's been mocked by some people, but it sort of helps. These are the so-called master regulators of subset differentiation. These are transcription factors that are absolutely required for either Th1 or Th2 or Th17 differentiation, or even Treg differentiation. And the one that is important for Th1 is called Tbet. It's part of a member of transcription family, transcription factors. And in fact, it's induced by gamma interferon stimulating STAT1. So the STAT1 basically causes transcription of Tbet. So we'll bring up these, I'll summarize all these when we go through each family. So you have these transcription factors, and they lead to active transcription of gamma interferon. And in addition, some more long-term, what we call epigenetic changes in the loci for these cytokines, so that they are in not a fixed state, but an active state that's passed down to daughter cell after daughter cell. So it's changes in DNA methylation, histone methylation, for instance, that are remembered as the cells replicate. Okay, for Th2 differentiation, I don't have to dwell so much on the definition of these things, but the categories are the same. There's a cytokine that's needed for the initial commitment, and the cytokine happens to be interleukin-4. This brings up the same problem that interleukin-4 is the end product, and where does the interleukin-4 come from? And this has been a real tough nut to crack, and there have been waves of interest in different sources of IL-4 that are considered to be the important Th2 driver. And each time they come up with a cell, somebody else debunks it in a prominent paper. But mast cells and eosinophils have both been implied based on papers that show that they can make IL-4, and if you somehow deplete usually these are mice, of these cytokines, you don't get Th2 differentiation. There is some evidence that the naive T cell can make a little IL-4 itself, and if you don't have the other signals, such as the IL-12 and the strong innate response, the IL-4 dominates. And it dominates by inducing its STAT6, and what's called the master regulator of Th2 differentiation, GATA3, and these begin to actively transcribe IL-4, and then you get an amplification mechanism. At the end of the day, you have a region of a chromosome that actually encodes for IL-4, IL-5, and IL-13 that's all in a very active state, and these are the cytokines that this type of T cell will make. And we'll talk about these functions briefly and refer later to Steve Galley's talk. And for Th17 cells, we have a similar paradigm, a similar set of events. The stimuli are probably extracellular microbes, such as various bacteria, Staphylococci, Streptococci, Candida, fungi. The dendritic cell is stimulated by them, and here some classic innate immune system cytokines are required. In mice, it's been clearly shown that IL-1 and IL-6 are involved. In humans, there's evidence for IL-1 and maybe TNF. And these are cytokines that are just possibly made even with no adaptive immunity. They're really important inflammatory cytokines for innate responses. But they alone do not drive Th17 differentiation. What is interesting is that the other signal that's needed, the other cytokine signal, is a signal from the cytokine TGF-beta, or transforming growth factor beta. And TGF-beta has always historically been, it's a ubiquitous cytokine made by all sorts of cell types, and it's historically been thought of as an anti-inflammatory cytokine. It prohibits or inhibits lymphocyte proliferation, and it's always thought to be something that would dampen down immune systems. And in fact, Abul, when he talks to you about regulatory T cells tomorrow, will mention TGF-beta's role in regulatory T cell development. That's an anti-inflammatory response. Yet here it is being very important for the driving of a very inflammatory cell. And the context is important. TGF-beta without these does not make Th17 cells. It actually will enhance regulatory T cell development. With these inflammatory cytokines, it drives Th17 differentiation. Again, there are transcription factors that are downstream of these, and for Th17 cells, this transcription factor, STAT3, is very important. IL-6 is important to generate STAT3. This cytokine called IL-23, which is also involved in differentiation, drives STAT3, and I'll show you an example of what happens when you don't have STAT3, but you might imagine one of the things is you don't get Th17 cells. The master regulator, if you will, the transcription factor that's absolutely required for Th17-ness, just for the phenotype and for differentiation, is something called ROR gamma T, a retinoid orphan receptor family transcription factor. And you can see that one of those cytokines actually induces ROR gamma T formation. So these cytokines that I mentioned briefly, IL-23 and I mentioned IL-21, are ones that are murky about what they do. Maybe IL-23 is important early on in human Th17 differentiation and in mouse later. I'll get to you in 1 second. IL-21 has been shown to be one of these autocrine amplifying factors, most convincingly in mouse, but again, that's controversial. And at the end of the day, you get a T cell that makes 17 and 22. Yeah? It depends on who you talk to. If you work on my floor where you turn the hall and you go down, and Vijay Kuchru, who's a prominent person in this literature, he'd say it's settled. And his colleague, David Hafler, who's now at Yale, are very much convinced that the TGF-beta story is settled. If you talk to other people, then it's not. And I don't really want to get into the details, but it was one of these things where people said that TGF-beta was important, then somebody came out with a paper saying, well, you don't realize that there was TGF-beta contaminating the serum you used. I think Abul's got an opinion here because he's standing up, but I think it's pretty much a very clear cut. We make Th17 cells in vitro in culture, and we know that TGF-beta is absolutely crucial for its differentiation. It's mouse cells.
It's not just the question was actually a very interesting question. Not just whether you need TGF-beta or not, because as Andy correctly said, that's been an argument. But some people say you need TGF-beta to turn on the transcriptional program of Th17 cells, and other people say that you actually need TGF-beta to shut off the Th1 and Th2 response because both of those inhibit the Th17.
Yeah.
So it's either directly inducing or it's providing a permissive environment.
And it's another part of the argument. I would also say that the only way we can analyze these are two ways. One is to put these artificial cocktails of these cytokines into a tissue culture dish with a naive T cells and see what happens. What do you get? And that's an approximation of in vivo, but a weak one. The other is to take a mouse and neutralize or knock out genes. You can't do the great experiment in human. And so the difference between human and mouse remains a problem. And so all we can do is see the experiments of nature in human. So the comment, and which I agree with completely, is even in this field, or as much in this field as anything, the difference between human and mouse is real. But I would point out also that nobody argues that there are IL-17 producing T cells that are important in human disease. They were discovered through mouse work, and 80% of what you've done in mouse is relevant to the human. But ultimately, there are important differences, so I would agree. Okay, so this is just the overall summary of all those names of transcription factors and cytokines. Here's your naive T cell. Here's your three pathways. TBET is the master transcription factor for this pathway for Th1, GATA3 for Th2, ROR gamma T for Th17. These are the stats that are involved downstream of the cytokines that drive these. These are the end result cytokines that are made. And I draw these awkward arrows to say that these cytokines can feed back and amplify the process. And so when we move on now, when I talk about these functions, I'll sort of assume you know all the details there. Yeah.
I had a question on the timeline. So do we know how much time it takes before the decision becomes final? Can we just go a little bit and then change the extracellular mix?
So the question is the timeline. How long does it take before you get down to these things? So again, in an artificial situation in vitro, within 4 days to 5 days, you can get well-differentiated T cell subsets from a naive T cell, which already is multiple rounds of proliferation. And there's very nice work from several labs about the importance of the synthetic phase and DNA synthesis and changing chromatin structure. In an animal, or more importantly in a human, where you really can get polarized subsets, I would argue that the evidence is maybe it's longer before you really see a phenotype that you can call Th1 or Th17. And some people argue there's always going to be a mix. So you can do this for single cells or a group of clone cells within a few days. Okay, oh yeah, there's the important cytokines. All right, so now we've got effector cells. We've got helper T cells that can theoretically do something. So now let's talk about this the effector phase. So I really sort of lied. I started back in the lymph node where Abul was because all that is happening in lymphoid tissue. And this is an important point. A lot of people will talk about TH subset differentiation, be looking at peripheral tissues, looking at the infected liver or the autoimmune brain. And that's not where TH subset differentiation starts. It may change there, but these events really happen with naive T cells. Okay, so once you get these effector cells, they get in the blood, they circulate, and they have to migrate to sites where they can do something, to sites of infection. So they get out of the blood, and the helper lymphocyte then is for most of our understanding, reactivated by antigen again. And this may be a little murky for people. For some of you, it's obvious, but others who don't follow cell-mediated immune responses, because it's not what you do, you may not get that. All that process I said before gets you a cell that is not necessarily, after the few days of activation, making those cytokines. It's just one that, when you reactivate it, it'll make a lot of those cytokines. And that's how we do it in the lab. I say I've made Th1 cells. It's not because they're actively making gamma interferon, it's because now when I re-stimulate them with antigen, they make a lot of gamma interferon and not any, say, IL-17. So these cells get out at sites of infection. They get reactivated by antigen presented by antigen-presenting cells. In this case, the antigen-presenting cells are not necessarily dendritic cells. They don't have to be, because effector cells don't require all those stringent things that only dendritic cells can do. Furthermore, dendritic cells are not necessarily migrating into these sites. Infected macrophages may present the antigen, and that's perhaps the most important. And then at this point, they release cytokines that cause the useful things that rid the infection. We'll ignore the CTL part of that for now, for a minute. So this is by way of review, and I'm going to brush over it really quickly, but Jason talked about how cells move around. Just remember that this is the time when effector cells get in the blood. They have to get back out to a site of inflammation. And in fact, what's going on in this complicated picture is what was summarized both by Ken and Jason this morning, that at the site of infection, there are cytokines and chemokines being made. The cytokines, such as IL-1 and TNF, make the endothelium sticky for leukocytes. They make them express things called selectins, like E-selectin or P-selectin. They increase the expression of adhesion molecules that bind to integrins, so-called ICAM1 or VCAM1. And the T cells that we're talking about will interact with these. They'll roll, they'll stop, they'll crawl through the endothelium, just like you heard this morning. And so the recruitment of Th1 or Th17 or Th2 cells to sites of infection require all these events, and therefore we talked briefly in a question from a previous lecture about you can target perhaps a immune response that's mediated by one of these cells by trying to block these adhesive interactions. And that's at least theoretically the way Tesabre works, that it's a integrin like VLA4 that Tesabre binds to one chain of, binding to VCAM and stopping the movement of a Th1 cell into the CNS. Okay, so that's just complete repetition, so I talked over that. So let's now dig down a little deeper and say, what do the T cells, whether they be Th1, Th2, or Th17, do when they're in the site of infection? So they've got out of the blood, and we'll use a generic cell here, but it's more like a Th1 than anything, and then I'll tell you in specific about what each of those subtypes does. Here's a lymphocyte that comes out, a T effector helper cell. And you should, by the way, this is jargon that I don't know if anybody formally defined, but when we say effector T cell, do you know what I'm referring to? Is anybody confused by that? Total blank. So effector is sort of the opposite of naive. Effectors are differentiated cells that actually do something. A naive B cell does not make antibody. All it does is be able to respond to signals and then ultimately become an effector B cell, like a plasma cell that makes antibody. And a naive T cell doesn't do anything useful other than respond to antigen, proliferate, and differentiate. It turns into an effector cell that actually does something useful. So that's what that terminology means. So helper cells are effector cells. Cytotoxic T lymphocytes are effector cells. All right, so the CD4 cell is going to see antigen again. It's the exact same peptide and the exact same NHC molecule that was on the dendritic cell in the lymph node, now being presented by a macrophage that's been infected. It recognizes the antigen, and what does it do? There ends up being this bidirectional interaction between the T cell and the antigen-presenting cell, where signals go both ways to ultimately lead to an effective response. I will refer tomorrow again to Shiv. We'll tell you about how this works when helper T cells interact with B cells, but I'm going to beat him to the punch for one of the molecules involved here. So an effector T cell, a CD4 helper T cell, will express cytokines. That's what we've been talking about. That's one of the things it does, that will bind to cytokine receptors on the antigen-presenting cell. So the antigen-presenting cell is presenting antigen, signals go down here, transcription leads to the production of cytokines, and the signals go into this cell. So two ways. In addition, effector T cells, certainly Th1 cells, will be induced, again by the signals from antigen recognition, to express a molecule that's part of this large superfamily of proteins called the TNF family, tumor necrosis factor family. Remember what TNF is. If you don't know TNF, then you flunked the innate immunity part of the course. So remember TNF. And TNF is only one of a large number of members that are homologous. They're virtually all trimeric proteins. And this one happens to be called, this brilliant name, CD40 ligand. It has a lot of information in it. Well, one of the things CD40 ligand obviously does is it binds to CD40. And what family of proteins do you think CD40 belongs to? The TNF receptor family. Okay. This is the TNF family, TNF receptor family. They also are trimeric. So this interaction, then CD40 is a terrific signaling molecule. Its downstream pathways are quite well understood. And CD40 signals synergize with signals from cytokines to activate this macrophage. I'll tell you what that process is, that activation, in a minute. And so this is the real useful part of a helper T cell response. This macrophage becomes activated, and presumably it does several things, one of which hopefully is to kill the microbe inside it. The others may be to help bring more soldiers into the site, more leukocytes to cause inflammation. Ultimately, you have to turn off immune responses. Every aspect of adaptive and innate immunity has as many regulatory parts to it as activating parts. And there are various ways that this is turned off. But one of them is, by absence of continued antigen exposure, this helper T cell is programmed to die. If it loses the signals that are being generated, including some autocrine growth factors, it will undergo an apoptotic death, and the number of cells will diminish. And you might sort of realize that you have to do this because these are dangerous cells to be around after the infection is cleared, because they make a lot of toxic things. Remember that bugs are still more like us than different from us, and what can kill a bug, it almost always can kill our own cells, so one has to have regulatory pathways. All right, so now I'll just superimpose now in our process of what we described of effector cells, the Th1, Th2, and Th17 story in these summary cartoons about what they do. So here's the Th1 cell that's derived from the naive T cell. Here's the interaction with the infected, or not necessarily infected, but APC that's phagocytosed the bug. It makes gamma interferon, and what it does is causes something called classical macrophage activation. And classical activation was just called activation years ago before somebody discovered an alternative pathway of activation. And what it does is increases various aspects of the cellular machinery for killing microbes here. And on a slide we have in a minute, I'll show you what they are. Now, this general cartoon was drawn, although we still draw it for new editions, before the advent of or understanding of a special type of T cell called T follicular helper cell, which is probably the important cell that is interacting with B cells to drive their differentiation to make antibodies. But we knew that cytokines would be very important in B cells making antibodies, and we knew, at least in mice, we knew which cytokines were associated with which type of antibodies. And so we have this general idea that Th1 cells, or at least gamma interferon, is important for B cells making certain types of antibodies that are really good at binding to microbes and then binding to phagocytes and being internalized. And they do that because there are types of antibodies that bind to something called an Fc receptor, which are receptors on phagocytes that bind to the end of antibodies, not the antigen binding end, but the other end, and bring them inside the cell. And it turns out that gamma interferon is really good at causing B cells to make a type of antibody that does this. In addition, there's this pathway called complement that nobody seems to want to talk about in this course because everybody hates complement because its nomenclature is so terrible. But complement is incredibly important for defense against microbes. And there are certain types of antibodies that activate complement. And again, interferon gamma is good at inducing those. The problem we have is this is well defined in mouse. In humans, we have much murkier idea of what cytokines are needed to cause B cells to make certain isotypes of antibodies. So we don't know necessarily whether it's such a neat story. Okay, now I told you I'd just delve into a little deeper about what those signals are that make the macrophage kill the bug. So I told you already about 40 ligand binding to 40 and gamma interferon, if this is a Th1 cell binding to interferon gamma receptor. Together, signal transduction processes lead to upregulation, that is transcription of the genes encoding for, and then ultimately secretion of a variety of cytokines, every one of which really ramps up the inflammation. Tumor necrosis factor, interleukin 12, interleukin 1, chemokines needed for the, remember, the activation of integrins and to drive the migration of these leukocytes. All these are enhanced by a combination of 40 signaling and interferon gamma receptor signaling. The generation of reactive oxygen species, basically free radical forms of oxygen that are really terribly toxic to bugs, is greatly induced by these signals. A macrophage that doesn't have any Th1 cell to help it will eat a bug, may generate a piddling amount of this stuff. Maybe there are some susceptible bugs, but others will survive. You need this extra kick from the Th1 cell in order to have enough reactive oxygen species, and among them including nitric oxide. So the enzymes that are needed to generate these are induced. Enzymes such as phagocyte oxidase and inducible nitric oxide synthase that make these. In addition, and we don't know really the relevance of this, these signals increase the expression of various co-stimulatory molecules, including B7 molecules. And why we don't know the relevance of this is, as Boole mentioned, we don't know how important co-stimulation is out here in the periphery, at least the B7 CD28 family. And so it's not clear whether, that's certainly a marker of activation, but we don't know its important role at that stage. Okay, this is just words saying everything I've just described, and so I'm not going to dwell on it. It's in your notes. Now, I mentioned this phenomenon of classical macrophage activation, and perhaps this slide, which is just a different version of something you have in your notes, but same information, is a little out of order because I haven't brought up alternative yet, but I'll describe these two general forms of macrophage activation to show you how Th2 cells come in. So the idea is that macrophages have a non-committed state, if you will, before they're in tissue. Macrophages, by the way, are constitutively present in just about any tissue in the body. Certainly, they can be recruited for the non-cognizant. That's not me. Good. What cells are recruited into inflammatory sites to replenish tissue macrophages or to increase the number of macrophages? Blood monocytes. Some people don't know that, but the monocyte is the circulating form of the tissue macrophage, and a blood monocyte gets into tissue and almost immediately changes its activity of what it synthesizes and becomes a macrophage. Nonetheless, it or the resident macrophages are not fully activated. In the presence of certain cytokines, they become activated. So I told you about gamma interferon already, generating all these responses. And so a classically activated macrophage is very pro-inflammatory. It's tissue destructive, partly because it makes proteinases, so-called matrix metalloproteinases of various forms that degrade extracellular matrix. So you can see, for a site of active acute inflammation, tissue is broken down. In part, it's because of these enzymes. That's what we've talked about already. Turns out that other cytokines, including the Th2 cytokines, IL-4 and IL-13, will make the macrophage into something that's called alternatively activated. And by the way, another nomenclature for this is M1 for classic and M2 for alternative. And alternatively activated macrophages, in large part, are characterized by production of mediators that increase the production of fibroblasts and collagen synthesis by fibroblasts, meaning that they create wound healing, or if too much of it, they cause scars. Or tissue remodeling in a destructive way. They also have some regulatory functions that may actually dampen this type of macrophage functions. For instance, the production of IL-10, which is a cytokine that is inhibitory to macrophage function. So there's this dichotomy. Like everything that you say it's one or two, these are oversimplifications. And if you look in the macrophage literature, you'll see there are 3 different types of M2 and more different ways of activating M1s. But that's all I want to do for these pathways. The point is, though, that this could be a healing process, but too much of it could cause disease, and there's a lot of interest in this. So Th2 cells will, in part, one of the things they'll do is they'll alternatively activate macrophages. and that will lead to enhanced wound healing, but also may lead to scarring. So there's a lot of interest, for instance, in lung disease, fibrosing lung disease, that could be due to chronic sort of Th2 cytokine-driven alternative macrophage activation. One of the important functions of IL-4, the signature cytokine or asignature cytokine of Th2, is to isotype switch B cells, to make B cells make the isotype called IgE. And again, I'll refer to Steve Galley and the importance of IgE in allergy. And so this is a very important pathway, and that we know is true in human and mouse. There's no question about the importance of IL-4 in this pathway. And then, as I mentioned, there's some of the same cytokines, IL-4 and IL-13, will increase mucus production in the gut and peristalsis, and you can imagine that's really good for eradicating worms that like to hang on to the mucosa. And IL-5, a classic Th2 cytokine, is very important differentiation factor and activator of eosinophils that have in their granules. Eosinophils circulate like neutrophils. They have granules stuffed with mediators, but they don't phagocytose. They release those granules, and including some really nasty stuff like major basic protein that can basically proteolytically degrade these thick walls that the worms have, and it's part of the anti-helminthic defense. All right, so I'm going to stop there for the functions of Th1 and Th2 and make some general statements before I move to CTLs. Maybe I'll prod you to ask a question when I'm done with the Th2, the CD4 cells, although interrupt me. There are some misconceptions that every year I think I'm going to pull this slide, but then I read a paper in the literature that just promotes these misconceptions. One is that, and you'll see this a lot in clinical literature, where everybody wants to assign diseases to either Th1 disease or a Th2 disease or a Th17 disease. They'll say that Th1s, which are important for cell-mediated immunity, and Th2 cells are important for humoral immunity. And in fact, now with the advent of Th17 cells, we know that you can't just say that Th17 cells are important mediators of immunity, cell-mediated immunity. But in fact, Th2 cells are not part and parcel really important for humoral responses. There are very few Ig isotypes actually driven by Th2 cells, IgE and IgG4, for example. And we know, as I told you, that gamma interferon is an important switch factor. But in fact, the advent of T follicular helper cells may make this even more of a dumb statement to make. The other is, and this started early on because the original definition of these subsets was made from mouse clones, and that attempts to try to find really well-defined subsets in human failed initially, that these subsets really only exist in mice and are not found in humans. And in fact, you can find very nice examples of cells that are individually polarized and strongly Th1 or Th2 or Th17, like in chronic exposure to antigens and chronic inflammation in tuberculous granulomas, for instance, in allergic lung. That's not to say that you might not find a mix of Th1s and Th17s in the same lesion, and that brings up another interesting question about the relevance of what one does or another. But there clearly are these subsets that exist. Yeah?
So if you don't mind, just a little bit about the point, but how come we keep saying, even immunologists, we refer to Th1 and Th2 as immune response? How come we keep saying perpetuating that if we know that's not the fact?
How come we do that? Well, we don't do that in textbooks and stuff. I think because it's so simple and it's such a dichotomy and it's easy to put in a one-line or an introduction to a paper or in an abstract, because if you have a finding that says something Th1, do something, that's the only reason I can say that. I think that it's the fact that Th1s are so dominantly important for macrophage activation and classic cell-mediated immunity that it left people saying, well, what do Th2 cells do? They knew they isotype switched for IgE. I don't know why, but it's wrong. Okay? There's no basis for it. I don't think any of the immunologists in this room would argue counter. So, they didn't take the focus class. Thank you. That's okay. The functions of Th17 cells. I've actually, I really didn't develop that yet. So I told you IL-17 and IL-22. Remember, IL-17 is sort of a mixture of at least two different cytokines in T cells. And IL-17 works on all sorts of cells. Just about every type of cell in the body has IL-17 receptors, and so there's ubiquitous response to them. On leukocytes and tissue cells, one of the important roles of IL-17 is to induce pro-inflammatory mediators. I've told you that already. Chemokines, tumor necrosis factor, IL-1, IL-6, colony-stimulating factors that work on bone marrow to make more of these. And so these all work together to increase inflammation, and in particular, neutrophilic inflammation. In addition, IL-17, as well, for instance, IL-22, induces epithelial cells and neutrophils to make what are called, I don't know whether Ken got into this, one of the mediators of innate immunity are these peptide antibiotics. There's a couple of families, defensins and cathelicidins, which are basically small peptides that get into microbial cell walls and kill the bugs. And they're made by epithelial barriers like the gut, like Paneth cells in the gut, or by neutrophils in their granules. And they're important defense mechanism against bugs. And IL-17 really enhances their production. So you can see that that's one way that they defend against microbes. IL-22 has a more subtle effect, but important. It actually allows epithelial barriers, again, like the mucosal barriers of the gut and the lung, to repair themselves. It induces mucosal epithelial proliferation and makes the junctions tighter. So it's important for keeping that barrier against the dirty microbial world. Okay, yeah. So the question is, could there be subsets that differ, and for instance, IL-22 dominant ones? And there is some literature, I think in the skin in particular, I've heard of that. And so it may exist, but there's no good molecular definition for it. But the problem is that if you look deep enough, and I'm not saying these are wrong, it's just that they're scattered studies, some are good, you can find people saying, here's an IL-22, not IL-17 producing T cell that in vitro does this, and vice versa. But yes, there may be subsets. That wasn't another question, right? There was a... Okay, so yeah, point is raised that, and in fact, in general, I should make the point, and in human it's more important, that not every immune response, and in fact, most of them don't fall into this actual clear distinction of Th1, Th17, or Th2. And I told you that these are most evident in human in chronic exposure to antigens. Okay, but let's say what are the rare human knockout examples that maybe can give you a definition of the importance of these things. So again, to try to find a disease where Th17s are knocked out would have to be maybe a ROR gamma T mutation. There are those in mice. I'm not aware of ROR gamma T mutations in humans. It probably was lethal. But there is this complicated disease called hyper IgE syndrome or Job syndrome, which has a lot of things going on because these kids have mutations of various sorts in STAT3. Remember, STAT3 was that transcription factor downstream of IL-6, IL-21, IL-23, all cytokines needed for Th17 differentiation. And these kids, the IgE part is a mystery, and it's not clear how it plays into the pathogenesis of the disease, but they suffer from multiple recurrent purulent infections with staphylococci, abscesses with candidiasis, so there's extracellular bacteria and fungi, and there's at least one striking study of impaired Th17 responses. If you look in the blood of a healthy individual, there's a certain percent of cells that make Th17 cytokine IL-17, but not gamma interferon. That's how you define a human Th17 cell, at least in the blood. And in these patients with Job syndrome, they make very little. So this is a sort of evidence, as indirect as it is, that Th17 cells are absolutely required for defense against these infections. You might argue that the STAT3 is needed for IL-17 production by other cells besides Th17 cells, and that's part of the basis of this. Okay, yeah?
So this is quite interesting.
Pardon me?
I'm saying that the data you are presenting is quite interesting. We are doing a similar study in malaria, and the results are opposite. Th17 appear to be increased to the highest in the category of kids with the highest mobility and
So the interesting question is a study of kids with malaria that the highest morbidity is associated with the most Th17 signature. And that, I don't know, I'm no expert in how that could play out, but in general, clearly, if you have a pro-inflammatory stimulus, but you still don't get rid of the bug, you can get disease. So tuberculosis, there's plenty of Th1 cells, and that's part of the disease. So it doesn't strike me as odd that in some cases, the microbe has evolved to even evade this type of defense, and it's actually contributing to the disease. A classic thing we teach in immunopathology for, I'm just going to switch the Mycobacterium tuberculosis, is the granuloma that's filled with Th1 cells making lots of gamma interferon, yet the bug has evolved to even avoid those classically activated macrophages, and the downstream is all these side effects. And so the increased inflammation, I don't know if this is in cerebral malaria or where you're looking, but I could see that a continuous stimulus for Th17 without eradicating the bug could certainly contribute. So it's needed for some infections, but some infections perhaps have evolved. It's interesting observation. Okay, so just as a point that is raised because there's a striking finding in a neat paper a while ago, where's our focus? Is Rick going to talk, Rick Blumberg? Okay, so we're going to have a great talk on mucosal immunity from Rick Blumberg in a day or two. And he will mention this idea that our microbiome, all the millions of bugs and the thousands of different species we have in our gut, really have a very big impact on what our immune system is and how it responds. And one striking example is, at least in mice, there's a particular bacterium, one species called segmented filamentous bacterium, where if you have a strain of mouse that's from one animal facility that has it, then you will get Th17 responses, not necessarily in the gut, but in lymph nodes, and get this disease in this particular model called a model of rheumatoid arthritis. And if you don't have that bug, you won't get it. And so one tiny little fraction of all the bugs in the gut of that mouse, whether it's there or not, is really, they're remarkable bacteria, but they're not that numerous, will totally skew the immune response in this model of rheumatoid arthritis. So whether you're autoimmune susceptible or not depends on one of these bugs, and the basis for it is whether you have Th17 cells or not. So I just point out that that's another important influence about how we skew our immune responses. Now, a question that is asked, and it's a good one, is are these subsets really fundamentally stable or not? And basically, we have to say, in large part, not. Although there is this commitment and epigenetic changes that make a Th1 cell really a well-defined Th1 cell, it can be changed to another type of effector cell, as well a Th17 cell. It has some important therapeutic implications because one could see that you might manipulate the cytokines in a way to try to cure a Th1 disease, and you may turn it into a Th17 disease, or vice versa. And even scarier is this fact that certain types of regulatory T cells can be turned into Th17 cells under the right conditions. And so there's a lot of interest in treating autoimmune diseases by transfer of regulatory T cells specific for some autoantigen. What would happen if, by accident, those Tregs, when they're in the body, actually turn to Th17 cells? The actual molecular definition for these plasticity is that we can look at certain modifications of the DNA that imply whether a genetic locus, and in Th subsets, the genetic loci are for the cytokines or their transcription factors, their master regulators. And a lot of work has been done with these, T-bet-GATA3 and ROR-gamma-T. And you look at histone modifications that imply, and I won't go into the details, that they're modified in this particular way, then you know this is a actively transcribed locus, or they're modified in another way and they're silenced. And we can see that in a well-defined, well-differentiated Th1 cell, you still have the ability to actively transcribe IL-17 and vice versa. So that's the sort of work that's been done, and there are at least evidence for all these arrows. Certainly naive T cells can go into all three, but Th17 cells can turn to Th1s, Tregs can turn to Th1s and maybe Th17 cells. There's not any clear evidence that I know about, last time I looked at this, about Th2 cells reverting into a Th1 phenotype or going there. Nonetheless, these are not well-defined, completely static end results for differentiation. I'm not going to talk about T follicular helper cells at all. Shiv is going to talk about it. I just say that there's this other subset, and so the more we look, the more we'll find CD4 positive helper T cells. Don't worry about this because Shiv will talk about these cells. They're very important in antibody responses. Somebody didn't start my clock, so I didn't start it. I was supposed to. Where are we at for time? It's 5, and I'm supposed to go till when? Okay. I'm going to skip recruitment, and you can look in the notes. The simple message is that if Th1 cells do something then different from Th17 cells that do something different from Th2 cells, you wouldn't want them all to come to the same place at the same time. I mean, we evolved these things to do different things. And if you look carefully, you can see that they have different adhesion molecules and chemokine receptors that drive them into different places. And that's the whole purpose of those slides there. Th17 cells have a chemokine receptor called CCR6 that binds to the chemokine CCL20, and that really is one of the important distinguishing features of why a Th17 cell might go into an inflammatory site. Th1 cells don't have that chemokine receptor. So for some reason, if some infection induces the expression of this chemokine, you'll get Th17 cells there and you won't get Th1s. So that's the general theme, but this is still a developing area. Okay. When I said earlier that IL-12 was a heterodynamic protein with 2 chains, I brought that out because there's actually an antibody that's used therapeutically that is specific for one of the chains of IL-12. Remember, IL-12 is needed for what type of differentiation? Th1. It's the first cytokine I said in all those morass of cytokines. So if you wanted to block Th1 differentiation, why not get an antibody to P40? There is one that's used and clinically approved. And it turns out that P40, lo and behold, is also part of the chain of IL-23. And IL-23 is a cytokine that's important for Th17 differentiation. I did mention that when I sped by. You'll see it in your notes. So in fact, if you're going to therapeutically block P40, you're going to actually get both these pathways. Well, you might say, well, that's probably a good thing because maybe these are both pro-inflammatory, they both contribute to autoimmune disease, and maybe they're both involved in IBD or MS, but one has to know what they're targeting. It turns out that there is another chain to IL-12, the P35 chain, and lo and behold, that's also on a cytokine called IL-35. IL-35 is actually made by regulatory T cells. So there was a lot of interest maybe in targeting P35 here to have a selective Th1 block. But in fact, if you did that, you may interfere with the way regulatory T cells work, and that's bad news. So in development of cytokine therapies, you got to know about these complex structures, these heterodimeric proteins, and what you're targeting and how they're shared by different proteins. And I won't go into it, but they're actually even shared proteins in the receptors. Okay, that's exactly what I just said in words. And I'm going to move to CD8 positive T cells. Can I ask for any burning questions or even non-burning ones for helper T cells before we move on? Yeah, you always ask the difficult ones. Is this relevant in humans?
The concept of plasticity, do you think, I just got interested in your opinion, do you think that these cells are a separate group of cells that are more plastic, or do you think we as immunologists have become obsessed with subsets and with the concept of plasticity we're going to have to let it go?
So it's a philosophical question, but it is this concept of the plasticity of these T cell subsets. Is there something special about them that make them particularly plastic, or as immunologists have we always sort of been too quick to put things in these baskets and be fixed? It's sort of a philosophical question. I think it's more of the latter. I don't think there's, I mean, you know, look at macrophages here. We have these subsets that can move back and forth. We have macrophages that can turn into dendritic cells and back and forth. I mean, that's a morass of a literature in terms of plasticity. So I think the latter is that we tend to want to simplify things, and that a truly terminal differentiated cell that can't turn into anything else is a rarity, I guess. Maybe a neuron in the brain. Yeah.
This plasticity, it could be for the microenvironment of the cytokines, perhaps.
So the question is, could this plasticity be due to the microenvironment of cytokines in a cytokine? Absolutely. I mean, that's one of the things that is thought. And there are mouse studies, at least, that say that, if you perhaps, for instance, IL-23 in the brain can change a Th17 response back to a Th1 response. So it depends on what the cytokines are. So again, cytokines can drive some of the change of these phenotypes. Okay, I'm going to move to CD8 T cells, and this is going to be a quick and inadequate job. And I'm not going to go through all the details of differentiation. It's all going to be on this slide right here. But remember, it starts with naive T cells. This time, they're CD8 positive T cells. A naive CD8 T cell coming out of the thymus is not a cytotoxic T lymphocyte yet. That's an effector cell. It's a cell that has the potential to become one. It recognizes peptide MHC class I. And in the presence, very importantly, of cytokines, interleukin-2 to drive proliferation, interleukin-12, absolutely essential for differentiation of an effector CTL. It will induce transcription factors that are akin to the transcription factors we talked about in Th1. In fact, T-bet and eomesodermin, that's part of the same family, are very important. And then you get, ultimately, the differentiation of cytotoxic T lymphocytes, which on this complicated slide is really this group here. And we'll ignore all these other things right now for lack of time. But the general idea, the one thing I want to ignore is migration. An effector cell is no good unless it can get to where it needs to go. So part of the differentiation of these cells is to induce receptors for chemokines and adhesion molecules. So you end up with a cytotoxic T lymphocyte. And so what does that mean? A cytotoxic T lymphocyte is a cell that carries a cargo that has granules in it that are needed to kill target cells and the ability to make certain cytokines. And so a cytotoxic T lymphocyte is like a helper T cell in that it can make a lot of gamma interferon, and it's like a killer cell because it's got lethal cargo in granules that can be released. And I'll bring that up in a minute. We're going to take one little sidetrack here because early in the lecture I said one of the things that CD4 T cells did is to help make CTL responses. And so the question is, how can this be? And it's been a tough thing to figure out. But one way that helper T cells can help in a CTL process is by actually the cytokines they make at the time of the initial naive CD8 T cell recognition on a dendritic cell. And remember, a dendritic cell can take up a microbe and can create peptide MHC class I ligands that can be recognized by this T cell receptor, but there's no reason why the same dendritic cell or a nearby one can, in response to the same microbe, find a different protein, generate a peptide epitope that loads class II and activate the CD4 T cell. They are not recognizing the same peptide. They're not recognizing the same MHC, but they are recognizing the same bug. And so the simultaneous activation of both can lead to cytokines that help promote CTL differentiation. And there is some evidence for certain microbes that you need CD4 helper T cells in order to get these CTLs. The other concept is that maybe the interaction, bidirectional interaction between the helper T cell and the dendritic cell will make this a better dendritic cell to ultimately activate the CD8 positive T cell. And the reason it might do that is because just like in the macrophage interaction, the CD4 helper T cell can generate CD40 ligand, and it signals CD40 in dendritic cells and makes these cells better expressors of co-stimulatory molecules. And so this is something that has been referred to as licensing this DC. The helper T cell gives the DC the license to activate the naive CD8 positive T cell. And so these are two theories about this. What I think is the general idea that is most consistent now, not shown in this slide, is that CD4 helper T cells, what they're really needed for is to allow CTLs to turn into or generate memory cells so that we have long-term CD8 T cells circulating in our body that remember a prior infection, and you need the helper T cell for that. So there is this important connection between 4 and 8, and I would say most of it has to do with memory generation. Okay, let's move to this exciting thing that CTLs do, they're killing, and talk briefly about how that does it. The first thing that happens is, now this is an effector CTL generated from a naive T cell. Remember Abul talked about clonal expansion? You have a viral infection, EBV infection, HIV infection at short periods of time after infection. You generate hundreds of thousands of cells from a single precursor, and a huge percent of your whole repertoire of CD8 T cells is recognizing one epitope of one virus. And every one of those cells is armed to kill, and for one particular peptide MHC complex. So this is one of them, and a target cell would be any cell that can be infected with that virus that initially stimulated this differentiation and expressed as class 1. So what cells in the body express class 1 MHC? Just about every cell except red cells and maybe a few in the brain, maybe a few in the placenta or something. And therefore, we have the potential of killing the reservoir of any type of virally infected cell. What happens is an immune synapse forms, just like what Boole talked about, with tight adhesion between the CTL and the target, using integrins on the T cell and their counter receptors, such as ICAM1 on the target cell. The antigen is recognized, and that causes the release of those preformed granules, the contents of which are within the synapse between the two cells, and that will ultimately, I'll tell you how in a minute, cause the death of this target cell. The CTL can actually release itself and not get killed by this process, get protected, and actually that CTL may actually go on to kill another infected cell. So that's the fundamentals of the process. You can see these very tight adhesions by electron microscopy between a CTL and a target cell. Here there are 3 CTLs in this system that's an antigen-generated one. Very tight apposition of the membranes. Here's a close-up. Here's the target cell that's going to be killed. There's the CTL. These are these granules, which are sometimes called secretory lysosomes. And this is the synapse, this tiny little cleft here. So very close apposition. And you might imagine, just like Boole talked about, I think, with cytokines, this allows the release of all those toxic granules into a very tiny space, and it's only going to affect the target cell and not bystander cells. And if you look by immunofluorescence, you can see here cathepsin is it's a proteolytic protein that happens to be in the granules of CTLs, and this is the CTL. They've localized right here between the two cells. This is the synapse. LFA1, that's the integrin. Notice it's on the outside. It's that ring that holds this whole two cells together. And talin is a cytoskeletal element that in the CTL has, again, localized right to this site. So this whole cytoskeleton of the CTL reorganizes to bring these granules to this one particular place and release them at that one site. So it's a very unique and careful and clean way of killing a target. It's truly a cruise missile targeting a particular site with no collateral damage like cruise missiles never have. Right. That was a political statement. All right. So what is the mechanism of killing? So I'll go through the contents of the granules of CTLs, but the big ones are granzymes. There are certain proteolytic enzymes called granzymes and perforin. And these work in conjunction with one another to cause the killing. And so the granules release these. They get into the target cell, and I'll tell you how we think they do in a minute. And they cause apoptotic death. Basically, what the granzymes do is they, several of them activate a proteolytic cascade mediated by caspases, which basically ultimately lead to the degradation of DNA. Lou. Is that you? Lewis? Yeah, you're here. Yeah, so in previous years, we never had the CTL lecture. So when you referred back to how we had it, now you can do this. So Lewis is going to talk about NK cells that use virtually the same killing machinery. So the contents of the CTL granules are as follows. These are specialized lysosomes, basically, biologically, if you look ultrastructurally. They contain perforin granzymes, a protein called granulysin, and this proteoglycan called surglycine that's probably something to hold all these things together in a way that doesn't cause the granules to self-destruct. And upon antigen recognition on a target cell, these granule contents, as I said, are released into the immune synapse. So I'll talk about these two first and return to granulysin in a minute. Granulysin is this protein that's a lipid-binding, highly cationic protein, and it has the capacity to lyse cells by both microbe and mammalian cells. And in addition, there's some literature that suggests that it's pro-inflammatory on its own without killing microbes. And we'll return to what granulysin may be doing after I talk about perforin granzyme. As I mentioned, surglycine, This is a proteoglycan that's ubiquitous in a lot of cell types with granules, mast cells, PMNs, endothelial cells, NK cells, and it's probably really serving as a packaging function. But the real important ones are the granzymes and perforin. So granzymes are serine proteases. They cleave a variety of substrates. I gave you an important one, these caspases, that in all cells in the body have this potential to kill themselves by apoptosis. They have to be activated, this cascade, and granzymes can do this. In humans, there are 5 granzymes, logically lettered A, B, H, K, and M. And there are 11 mouse granzymes. And it turns out there's actually some polymorphism in some of these. So when we study granzymes in mice, we may not be reflecting the full function that may be in humans. Granzyme B cleaves target proteins like caspases after aspartate residues. It's got the strongest of all these for apoptotic function, and it's most clearly implicated in the way CTLs and natural killer cells kill. And by knockout studies, if you have a granzyme A knockout or a B knockout, there is variable resistance to some microbes and tumor challenge. A double knockout is more susceptible, so granzyme A has some role in protection. But I point out that it's not really as strong a phenotype as you might think. It's actually when you get to perforin that you see the real strong phenotype of a lack of true CTL function. So perforins are pore-forming membrane disruptive proteins. They're like the complement that none of us will talk about but keep referring to. The terminal component of complement C9 is a polymerizable protein that forms pores and membranes, and perforin is in some way structurally similar to that. So perforin knockout mice are very susceptible to a whole variety of tumors, models, and viruses. And this is the strong phenotype that you'd think if you didn't have any CTL activity. And it's clear that perforins are needed to get granzymes or the granule contents into target cells. How this happens has been an amazing and frustrating mystery since it seems like such an obvious thing to figure out, but it's been a struggle, and I'm told by the experts that why it's a struggle is the microenvironment of that immune synapse is something that you cannot really readily replicate in an in vitro model very well, and it's hard to know what's going on there. So one idea is if perforin's a pore-forming structure, it can form a pore, and the granzymes can go through the pore. And there's some problems with that because of the selectivity of the pore and what gets in, and it doesn't make sense in terms of all the data. The other idea is that granzymes and perforins are actually endocytosed in a membrane-bound endocytic vessel into the cell, and then perforin has a secondary role of breaking up or disrupting the endosome membrane and allowing the perforin to get out. And it's probably this latter model, there's some version of it that's true, and this is one recent paper from Judy Lieberman recently that at least the pictures are pretty and shows the idea. So that if you have granzymes alone, which are genetically fixed or stained to be green fluorescent, you can see that when a CTL gets to a target cell, these granzymes don't ever cluster, and you don't get any apoptosis of the cell. This is 20 minutes. If you add perforin, you get these large collections of granzymes, and then they cluster near the nucleus, and you can see them right around the nuclear membrane, and then you end up with an apoptotic nucleus. And it shows that perforins are absolutely required for actually the delivery of the granzyme into the cytosol of the cell. And what is thought is happening is that perforins will perturb the target cell membrane. As part of the reparative process, the membrane will swallow up both the perforin and the granzyme. And then, once again, the perforin is in the endosome and will disrupt the endosomal membrane and allow the granzymes to get out. So that's the best I can do, at least, in explaining how you need perforin, but you absolutely need it for CTL function. So just mentioned that there's other ways that a CTL possibly can kill a target cell by death receptors such as Fas and Fas ligand, which I won't go into, but these are, again, members of the TNF family and TNF receptor family that have signaling. The Fas downstream has signaling that induces apoptosis. And there's some evidence that we know that activated CTLs have Fas ligand. Mouse models don't show a major role for this pathway for death. Okay, so that's how CTLs kill target cells. Can CTLs actually directly kill microbes? And it turns out that there's really an abundant literature in various places that say yes, including bacteria, they're mentioned here, parasites, and fungi. It does require the CTL priming, so that they have to be of activated CTL, but it does not require MHC-dependent contact like I've just described for granule exocytosis. And the killing is mediated primarily by this substance, this enzyme called granulysin, delivered by the granule pathway. And you can find pretty pictures of conjugates between CTLs directly and microbes, not mammalian cells. Obviously, this wouldn't be an MHC-restricted recognition process. So there's probably some important role for the direct killing of microbes by CTLs. So what happens when you don't have CTL function? And what is the human knockouts? And I put the asterisk here because this particular interesting disease, you not only don't have CTL killing, but you also don't have NK killing. And basically, this is familial hemophagocytic lymphohistiocytosis, or HLH. And there's lots of mutations that cause this devastating disease, but many of them are perforin gene mutations or genes encoding proteins that are actually needed for granule exocytosis, such as RAB1, for instance, or the syntaxin. And what happens in these people who cannot mount a good CTL or NK response is they get uncontrolled activation of both 4 and 8 T cells, largely by cytokine production. They lead to abundant macrophage activation and proliferation. And these macrophages eat our own cells, or eat the patient's own cells. That's the hemophagocytic component to it. And this is basically a disease that is a overreaction because of the lack of being able to control infections through the CTL killing. In fact, perforin knockout mice infected with certain viruses develop an HLH-like syndrome as well. So you can't say that's just CTL, but I think in large part it is, although Lewis may argue that maybe it's the NK cells. What happens with a more global CD8 deficiency? Well, not just perforin. There are rare examples of this, and they're not perfect. So there's certain very rare mutations in the CD8 gene itself, in the transporters of TAP1 and TAP2 that are needed to get the peptides into the endoplasmic reticulum, the ones that are generated by the proteasome and class 1 pathway. And obviously, if you can't make CD8 or you can't make class 1 MHC proteins on the surface because of these mutations, you will be able to activate CTLs. And there are CD8 deficiencies in these. And they get sinopulmonary infections. Interestingly, a lot of bacterial ones that you wouldn't think from a lot of literature that CD8 T cells would be important. DOC8 is another rare deficiency that's more complicated, but there's a CD8 deficiency in that, as well as B cell deficiencies. And again, there's some severe viral infections. The bottom line is that these various immunodeficiencies show that CD8s are important. It is important as we end up to point out that CTLs, as important as they are for protection, are also the cause of a lot of misery. They play a role in a lot of pathogenesis of disease. There's a good literature on the fact that perforin is actually permissive for malaria. It's sort of like your question about malaria before. How can a T cell that's supposed to protect against infection contribute to the infection? And here's an example in that the perforin is actually important in causing the killing of brain endothelial cells in cerebral malaria. And perforin clearly plays a role in a lot of autoimmune destruction. It's involved in the killing of beta cells in the pancreas and type 1 diabetes in the early stages of the disease. CTL are major contributors to post-viral myocarditis that ultimately will lead to dilated cardiomyopathy, very important in allograft rejection, and in the hepatitis caused by HBV and HBC. Hepatitis V, C, whatever. Okay, yeah. What if you took away LFA1 and LFA2? So remember that not only is LFA1 important for immune synapse, but it's important for getting leukocytes, all leukocytes, to sites of inflammation. And
Yes, you would have to take them away after you had your receptor. That would be very hard to.
you mean take them away as a therapeutic?
I don't know. I just.
There is a. What I was going to say.
If you didn't have it while the infection itself would do some effect, would you still have the crucial
So your question is, how important is the LFA1 in that immune synapse formation, as you're saying? And it's clearly important. In vitro studies, early ones from Tim Springer, who basically found these molecules by looking for cell surface molecules, could show that you could block the conjugate between a CTL and a target cell by blocking antibody against LFA1, and you got no conjugate and killing. So LFA1 is actually crucial for that interaction. Does that answer your question? With the microbe? That's a good question. You mean like the mechanism that I said for granulysin killing the microbe? I have no idea. But they won't have the ligand for LFA1, so I don't know. But we don't know how important that component is in defense. We have some evidence, but we don't. But you're right that the conjugate of a CTL with a microbe would be very different than a conjugate of a CTL with a target cell. All right, finally, I know it's really late, but I just have to, I'm supposed to just mention memory T cells. At the end of T cell mediated immunity, hopefully you have generated cells that last for a long time, that are there to generate what we call a memory response. The characteristics of memory T cells, and a lot of these are also characteristics of memory B cells, are they have a prolonged survival, and you don't need the antigen. So the infection is cleared, but you still have lymphocytes in greater number than you started with before the infection, and more rapidly able to respond. And so they are rapidly recall type T cells that have a long lifespan. They are actually functionally silent until they're reactivated. In some sense, their subset lineage is remembered. There's some evidence that there's Th1-ness and Th17-ness in memory cells. The markers of memory cells are that they have the receptor for IL-7, which is a cytokine that's important for quiet cells to live, even naive cells. In humans, memory cells have this isoform of CD45. That's the phosphatase that Boole mentioned in T cell activation and regulation. And they have the RA form. RO, that's wrong. RO. Cross out the A in your notes. It's RO. Memory RRO. Sorry about that. Now, it turns out that there's a large literature in both human and mouse that there are two general families of memory cells in T cells, so-called central and effector subsets. And the ones that reside out in the tissue, for instance, in the mucosa of the gut or the lung, are what are called effector memory cells. And they have a phenotype that is the opposite of the phenotype of naive cells. That is, they don't go to lymph nodes. So they don't have L-selectin, and they don't have CCR7. These are the molecules that Jason talked to you about that are needed to get a naive T cell in a lymph node. A effector memory cell doesn't have these, and they therefore don't recirculate into lymph nodes, and they're found in mucosa. And they respond rapidly by producing a lot of gamma interferon, for instance, in the periphery, and rapidly provide some defense on re-exposure. In the lymphoid organs, there are memory cells called central memory. They do have the molecules that allow migration into the lymph node. So naive cells are not the only ones that go into lymph node, this subset of memory cells. And they respond by proliferating rapidly, so sort of re-expanding the clone of cells against a particular microbial antigen. And there's lots of evidence for these in mice and in humans. Most of the time, you can only probe these in the blood, but there's some interesting, nice work in the skin now that shows that there are effector memory cells that basically hang out in the skin for a long time. They don't even recirculate, and they're there to respond and defend against microbes. How these are generated, whether they're generated as a separate branch from the actual effector cell or in series with them, is not clear, and I'll just end with that picture to show you the conundrum. That you have a naive cell turns into an effector cell. Well, maybe some surviving part of this, remember this large expanded clone will shrink, as I said, as part of the regulatory component after an immune response occurs, and these may be the survivors. But it may be that there's, in fact, a separate pathway for the memory cell differentiation that really never intersects with or is directly a descendant from these effector cells. And so with that attempt to try to end before dinner is over, I'm going to stop. So, are there any questions? Probably I don't know whether anybody's going to have tolerance for these. Do all effector T cells express CD40 ligand? You know, I'm embarrassed to say that I we've stained CD40 ligand on what we call Th17 cells, but I don't know how pure they were. Th1 cells clearly do. I'm not aware that Th2 cells do. Who's the Abul? Do Th2 cells express CD40 ligand? What's that, Shiv? Yeah, well, according to Shiv, all activated CD4s do. So the answer is yes. All right, enjoy the
Okay,
margarita.
couple quick announcements. Dinner is